Hi, today our group will present about case study Mus musculus or house mouse. Hi, my name is Nur Adila binti Sakaria and my matrix number is E2483462. Now I will explain about what is Mus musculus. Mus musculus is commonly known as the house mouse. It's a small rodent that is widely distributed around the world. Its preferred scientific name is Mus musculus linnaeus. 1758. This species is assigned the EPO, which is European and Mediterranean Plain Protection Organization, which is called MUSXMU, which is used for standardized identification in pest management and scientific database. House mouse or Mus musculus, region native to Eurasia but introduced worldwide through association with humans. Highly adaptive, the house mouse has both behavioral and physiological traits such as the ability to survive in buildings and aboard ship, a tendency to move into an agriculture field and live when the habitat change, and a rapid rate of reproduction that allow it to thrive wherever humans do. And now this is a physical characteristic of the house mouse musculus, color variation by type, and size and other features. Next is chromosome structure of mus musculus. Firstly, chromosome number in the mus musculus or house mouse. Mus musculus or the house mouse is a widely used model organism in the biological and genetic research. Second, a key scientific advantage is its well-characterized karyotype. For the chromosome details, they have a diploid chromosome number, which is 2n equal to 15, which is have a 90 pairs of autosome and one pair of sex chromosome. So XX in female and XY in males. Most chromosomes are telocentric with centromeres near one end, giving a rod-like shape under the microscope. The X chromosome is a large and easily identifiable. The Y chromosome is small and compact. And you can see the figure 1 for show the karyotype service for mouse cell, and for figure 2 most musculus, which is nuclear for chromosome for house mouse most musculus.
Welcome back, friends. In Gene Allen School, we learned about the basic of genetic engineering, biotechnology, and molecular biology. I hope every lectures are very useful to you to know about the basic of genetic engineering. So today we will learn about one of the most important topic of genetic engineering, and that is the mouse genome and the human genome. So you know that human genomes, everybody know about the human genome, but you know that in laboratory we normally use with the genome of mouse, because human genomes are not available to use, or there is some ethical manners, so that the mouse genome are used in the genetic engineering laboratory or for the research work. So we have a question, that is why mouse genome are used instead of human genome or other type of genomes. Why there is mouse? So there is some specialty obviously having the mouse genome, so that we can learn about, or we can research with the mouse genome, and you will get the result that will compare to the human genome. So we use the mouse genome. So
Number of genes of mus musculus. The article was provided for research purpose. Number of protein genes for mus musculus is 22,428. Number of RNA genes is 3,697. Number of genes of musculus. Less than 1% of the approximately 30,000 100 protein coding genes in men and mice have no autolog in the other species. There are 1,200 new genes in the list of predicted mouse and human genes, many of which are linked to illnesses in humans. Many of the genes predicted by the computer might be Phytogenesis according to manual inspection, enhancing the identification of genuine no-coding RNA genes and tRNA gene sets was another benefit of comparative analysis. Number of genes of musculus. Number of protein coding is 22,198. Number of non-coding is 17,661. Number of transcript physodogenes is 506. Number of non-transcript physodogenes is 9,846. Number of genes of musculus. At the nucleotide level, mouse sequences could be aligned with about 40% of the human genome. The authors hypothesis that during recent evolution, both genomes have experienced significant deletion. According to analysis, evolu- evolutionary selection affects about 5% of the genome, which, which is significantly more than just the coding regions. Non-coding RNAs and regulatory sequences may be found in these areas, with significant variation across chromosomal regions. The neutral substi- substitution rate is estimated to be roughly of a nucleotide subs- substitution per site.
Hi, my name is Muhammad Danis bin Muhammad Izailan. My metric number is E24A3425. In this video, I'm going to explain about the useful of Mus musculus gene in agriculture. Mus musculus is an important organism in field of agriculture. This is because Mus musculus is often used in field of biotechnology and genetics. One of the reasons is that Mus musculus has many genes that can be used in a field of agriculture. One of the gene is leptin-lep gene. Leptin-lep gene is located on chromosome 6, which produce the hormone leptin. Leptin regulates appetite and energy metabolism. Mice with mutation in this gene, known as op mice, become obese and have metabolic problems. The study on op mice helped to understand how livestock storage store fat and how nutrition can be optimized for livestock growth. The next gene was growth hormone genes. The growth hormone gene was located on chromosome 11 in musculus. It produced growth hormone MGH, which is important for muscle formation and metabolism. Studies on transgenic mice carry this gene have shown significant increase in body size and muscle mass, and the result of these research have been used in development of farm animals such as pig and salmon that grow faster, thus helping improve growth efficiency through biotechnology. Now I'm gonna explain the case study of musculus gene in agriculture. The first gene was leptin lep gene. The case study was association between leptin gene for polymorphism and growth traits in Bali Center. This study aimed to identify molecular mark of the lep gene and their association with growth train based on SNPS in Bali Center. The result of the study showed that SNP G.2913C T had a significant relationship P less than 0.05 with various animal growth characteristics indicate the role of leptin lep gene in animal growth. The next gene was growth hormone gene. The case study was high diversity of the chicken growth hormone genes and effect on growth and carcass strain. These studies say that the chicken growth hormone CGH gene plays an important role in regulating growth and metabolism, leading to potential correlation between CGH polymorphism and economic trend.
Hello, my name is Alif Shahmi bin Abdul Malik Al Faisal. My metric number is E24A350C. I will explain some of the genes and protein product of the organism capability that have been used in pharmaceutical. In the pharmaceutical field, genes and protein products are very important because they help in the production of biotechnology-based medicine that are more effective and safe to use. Here are some genes and protein products of the ability of organisms that have been used in pharmaceutical. The number one is P53 gene. Function: P53 is a tumor suppressor gene that is important in regulating the cell cycle and apoptosis, controlled cell death. Protein product: the P53 protein. Pharmaceutical uses in animal models such as P53 knockout mouse. When this gene is deleted or mutated, the animal are more likely to develop tumor. This makes them ideal for detect the carcinogenic effect of drug in short-term testing. Number 2, ionobiotic metabolism enzyme CYP450 genes. Function: These genes control enzymes responsible for metabolizing drug and foreign chemical in the body. Protein product cytochrome P450 CYP enzymes. Pharmaceutical use organisms, usually mouse, that have been modified to express human form of CYP enzyme are used to study drug metabolism and identify toxic or carcinogenic metabolites. The last is MDR1, multi-drug resistant 1 gene. Function: control the transport of drug and toxin out of the cell. Protein product P-glycoprotein. Pharmaceutical use: animal models carrying the human version of the MDR1 gene are used to study drug absorption and resistance, important in the development of cancer treatment and pharmacokinetics.
Hello, my name is Nofazira and I'm going to present about method and critical analysis. When studying the P53 gene, scientists follow a very careful set of steps, especially when it's come to understanding cancer and testing new drug. This is where the magic begins. Scientists create special mice known as genetically engineered mouse model (GEMMS). This is a very precise test. They design a small piece of DNA with specific marker around the P53 gene. This piece of DNA is then put into very early mouse cells, embryonic stem cell. After careful check to ensure the gene has been changed correctly, these cells are placed into mouse embryos. The resulting mice are then bred until they have the desired P53 change throughout their body. For even more control, scientists can make conditional P53 mice where the genes can be switched off only in certain body parts or at specific time. This helps them study exactly how the gene affects tumor growth in different situations. Next, to test new drug, scientists implant human cancer cells, sometimes even real tumor piece from patients, into mice. These mice have weakened immune system, so they don't reject the human cell. They carefully inject the cell under the skin or into an organ, letting a tumor grow. Then they measure the tumor size using small ruler or a special light system. If the cancer cell glow. Once tumors are ready, mice are divided into groups for treatment. They receive the new cancer drug, like Nuclein 3A, through various ways, by mouse injection into the belly and or directly into a vein. They carefully track the tumor growth and the mouse weight and help to see if the drug works and if it causes side effects. After the study, the tumors are removed. Scientists then use powerful technique to analyze the protein and gene inside. Western blotting helped them measure level of P53 and related protein like MDM2 and P12, showing if the drug successfully activates P53. Immunohistochemistry used collect TET to visualize P53 and other protein directly within the tumor tissue under a microscope. They also look for a sign of cell death to confirm the drug effectiveness. Critically, the extensive use of P53 mouse model have revolutionized our understanding and treatment of cancer. In genetic and molecular biology, this model have allowed for the precise elucidation of P53's intricate molecular pathway, its interaction with another protein, and its tissue-specific role in tumor suppression, providing fundamental insight into cancer biology. From a medical perspective, they have a significantly accelerated preclinical anti-cancer drug testing, leading to improved prediction of drug efficiency and patient response. This have substantially de-risked drug development, guiding clinical trial design and contributing to the development of more targeted and personalized cancer therapies. The second gene is CYP450. A major step is creating special humanized mice. Scientists use cutting-edge gene editing technique to swap the mouse on CYP450 genes for the human version. For example, replacing the human versions, the mouse version of the CYP3A with the human CYP3A4 genes. These humanized mice then produce drug much like a human would. Researchers give the experimental drug to these humanized mice. At different times after the dose, from minutes to hours, they collect small blood sample. Using highly precise machine called liquid chromatography mass spectrometry, they can measure exactly how much of the drug and its broken down product are in the blood. This gives them a detailed drug journey map, how fast it absorbs, where it goes, and how quickly it's eliminated. This study can also help predict if a new drug will interfere with other medication. By giving mice a known drug, like an antibiotic that also uses CYP450 alongside the experimental one, scientists can see if the CYP450 enzyme get overloaded or become less active, leading to dangerous drug build-up. Beyond blood tests, scientists can take liver tissue from the mice, isolate the enzyme factory microsome, and test the activity directly in a test tube. They also use qPCR and Western blotting to see if the amount of human CYP450 gene and protein are normal or changed by the drug. For critical analysis, these mouse models have dramatically improved our knowledge on how our body drug processing factories work, including how their genes are turned on and off. They help drug companies predict drug interaction and how fast drug will be processed in humans, leading to safer dosage and preventing harmful side effects. This means fewer surprises when a new drug goes into human trials, making drug development faster and more reliable. Last but not least, the MDR1 gene. The MDR1 gene makes a protein called P-glycoprotein, which acts like a powerful cellular pump. Its job is to push drug and toxin out of cell. Scientists create special mice to understand P-glycoprotein role. This include MDR1 knockout mice, where the P-glycoprotein gene is deliberately turned off. These mice allow researchers to see what's happened when drugs aren't pumped out. They can also create transgenic mice that produce more P-glycoprotein, helping them study how drug resistance develops. A key part of the research involves giving a test drug, which P-glycoprotein normally pump out to both regular mice and MDR1 knockout mice. At various times after the dose, the mice are humanly put to sleep. Scientists then carefully remove organs like the brain, liver, or kidney and precisely measure the drug concentration in this tissue using LC-MS. If a drug builds up more much in the brain of a knockout mouse compared to a regular mouse, it strongly indicates that P-glycoprotein normally pump that drug out of the brain. This study also vital for understanding drug safety. If a drug is usually kept out of the brain by P-glycoprotein, giving it to the MDR1 knockout mouse could lead to a very high level in the brain and cause harm. Scientists carefully watch these mice for signs of neurological problems like shaking, clumsiness, or unusual tiredness, often using standardized scoring system. Specific behavior. Last but not least, for the critical analysis, this mouse model has been key to unraveling the complex way drugs move in and out of the cell and how multi-drug resistance works. They have shown us how P-glycoprotein protects different parts of the body, not just the brain. This knowledge directly impacts how drugs are designed. Scientists can now create drugs that P-glycoprotein won't easily pump out or develop ways to block P-glycoprotein to make other drugs more effective, especially in fighting drug-resistant cancer or delivering drugs to the brain for neurological conditions. This means safer and more effective treatment for patients. Conclusion: to wrap it, using advanced mouse model to study genes like P53, CYP, 450, and MDR1 is a huge part of a modern drug discovery. The detailed step from cleverly designed mice to carefully testing drug and analysis result in the lab have not only taught us fundamental things about gene and cell, but also given us mental information that directly translate into new treatment. These mouse models are powerful tools for testing new drugs, spotting potential danger, and developing methods smarter for effective therapies. With ongoing innovation in mouse research, we can look forward to even more amazing discovery as we continue the journey to find safer and better medicine for everyone. That's all from us. Thank you.
I think that's all from us. Thanks for your attention. Bye.